Yeah, hi, I'm Lyduine and Sophie. I'm a postdoc researcher at the Amyloid-β project, where I also do some scientific coordination and project management. And actually also done my PhD on the Amyloid-β project focused on amyloid-β and specifically regional amyloid-β.
Hi, my name is Sophie Mastenbroek, and I've been part of the Amyloid-β team for quite some time now, actually, from 2020 onwards. I did several internships with Amyloid-β during my master's degree in biomedical sciences. And since September of last year, I started my PhD at the FUMC in Amsterdam, which is a joint Amyloid-β biofinder PhD. And my focus is on heterogeneity in Alzheimer's disease.
Yeah, there's also work that was really a focus that I had with my colleague Isidora Lopez-Alves for a while that we wanted to do. Within the context of Amyloid, I've assessed many different amyloid PET scans, and actually I'm a certified reader myself. And while assessing these scans, I saw there were clear patterns in the scans. Not everyone had the same point of origin where amyloid pathology starts to accumulate. So we wanted to see if we can, with data-driven manner, identify subtypes into the accumulation pattern that we see in these scans. And for this, we wanted to apply a data-driven method that was developed by UCL, which is called the Subtype and Stage Inference model, or SUSTAIN. And we applied that method to over 3,000 amyloid PET scans that we got from different cohorts at the FUMC, but also open access with a collaboration, for example, with the Barcelona Beta Brain Research Center, the Alpha cohort. To see if we could find different subtypes in this model. And interestingly enough, we found exactly the ones that I was hoping for to see. So I've been always saying, looking at these scans, you see some subjects that have frontal pathology first, subjects have parietal pathology first, and a subset that has occipital pathology, which is often actually circumvented in amyloid PET research. And exactly those three subtypes emerged from our data. So one that started frontally, one that started parietally, and that one that started in the occipital lobe. Yeah, so what this model can do is that it takes basically cross-sectional data from all these subjects and puts it into a longitudinal trajectory. So what we saw is that we found these three subtypes of accumulation pattern. Most people were assigned to the frontal subtype, so that was the most common one. And also that was related to most subjects having ApoE4 carriership. Then we had the parietal subtype, which was the second most common subtype. These subjects were generally younger and about the same in terms of ApoE4. And then for the occipital subtype, it was interesting that we saw there was overrepresentation of dementia cases. And when we did a post hoc analysis in one of the cohorts that we had more information on the clinic, we saw that this subtype is overrepresented in cases of DLB. So there might be some co-pathology being picked up with the occipital subtype. So cross-sectionally at this point, this was a cross-sectional paper, we saw some clear differences in risk factors between the three subtypes that we identified with SUSTAIN. So this was cross-sectional work. So the follow-up, what we really are interested in, what are actually maybe downstream effects of having these different subtypes in terms of accumulation of amyloid pathology. Do we see that subjects have a different pattern or burden in tau PET or in atrophy, maybe in clinical decline? So the follow-up work is something that we are focusing on at the moment. And actually that was what Sophie is doing within her PhD project.
For the next steps, I'm currently working, as Lyduine mentioned, on a follow-up project about the amyloid PET subtypes. And I think a very important first next step is to validate these subtypes in an independent cohort. So in other words, can we also identify these three subtypes in unseen amyloid PET data? And second, we're currently investigating whether these three subtypes show clinical relevance. So whether they differ on baseline and longitudinal cognitive performance. Atrophy is measured by MRI, and tau pathology is measured by both CSF and PET. And for now, we've looked mostly at the cognitive data. And here we did find a significant difference between the three subtypes, both at baseline and over time. And specifically, interestingly, the occipital subtype seemed to have a lower performance in specific cognitive domains compared to the other subtypes, whereas the data suggested that the parietal subtype actually showed a higher performance in certain specific cognitive domains. So we do see definitely some differences between the subtypes in terms of cognitive performance. So now it will also be very interesting to see whether these subtypes also differ on atrophy and tau measurements.
Yeah, for me, that was actually really at the beginning of this project. So as I stated at the beginning, I've seen several hundreds, maybe close to 1,000 PET scans, and I saw these patterns, and I was basically wanted to see if we could find it with a data-driven method. And first time running Sustain and getting the results, I was actually on holiday. But my colleague Isadora texted me, you have to see this, because we found exactly the 3 subtypes that we were hoping to find. So for me, that was really a very interesting moment that we could find something that we see with the human eye, also in a data-driven manner. For me, that was an inspiring moment to continue with this project with a very strong focus.
So for me, the most inspiring moment during this project was to see how much data is currently available. Because for these kinds of machine learning projects, you ideally want to have a very large data set, of course. So I was very impressed to see how many different cohorts currently have amyloid PET data available for so many participants, and also that the field really works together to share this data with each other. So it was really inspiring to me.